This video is going to be a review of cellular injury, all the way from reversible through into irreversible. So we have our cell, nice round shape. Within our cell, we have our nucleus. My attempt at DNA, that's our nucleus. We have mitochondria, the energy powerhouse of the cell. We have endoplasmic reticulum, covered in protein-making ribosomes. And this cell has a blood supply. All of the cells in our body need blood to be flowing by for the delivery of oxygen to be used in aerobic metabolism, the formation of ATP using oxygen in the mitochondria. We also have glucose and important energy substrates floating in the blood that get delivered to the cell. Okay, so with hypoxic injury, something is preventing the cell from getting its normal amount of oxygen, so the cell does not have enough oxygen to meet its needs. So right away, the mitochondria is under stress because it doesn't have the oxygen required to accept the electrons of the electron transport chain, so it can no longer make ATP effectively. For those cells that have glucose stored in the form of glycogen, they have a little bit of time, can buy themselves some time, because glucose out in the cytosol can actually produce ATP anaerobically, which means without oxygen. So we can get a little bit of ATP formed if there's glucose around. The end result, though, ends up with lactic acid being formation. And as the name implies, acid is going to lower the pH of the cell. That in and of itself isn't terrible, but lowering the pH, our enzymes operate within a really narrow range. So a lower pH can make abnormalities in enzymes within the cell. It also gives a really important visual in that when the pH is dropping, you get what's called chromatin clumping, which is the genetic material kind of clumps up. It doesn't have a lot of significance to the cell outside of being a visual marker that the cell is under stress. So we continue to not have ATP, we continue to not have oxygen delivered to the cell. And why is oxygen and its ATP products so important? We have tons of processes within the cell that require ATP. And a very big one is the sodium-potassium ATPase, which creates this big gradient of a bunch of sodium outside of the cell and very little inside the cell. Oops, drawing potassium out there. So when we cannot deliver blood to this area or enough oxygen, we can't maintain the ATP necessary for the sodium ATPase to be functional. If we can't keep pumping sodium out and there's a ton of sodium outside of the cell, it will leak into the cell. So this sodium will move down its concentration gradient and enter the cell. The significance of that step is that water always follows sodium. Where sodium goes, so too does water. As water enters the cell, we get cellular swelling. The whole cell gets bigger as it fills with water. So we see cellular swelling. This fluid can move into the organelles as well. We can see swelling of the mitochondria. Really importantly, these ribosomes actually float off of the swollen endoplasmic reticulum, which decreases our ability to make new proteins, which is going to be really critical, especially as the cell is under stress. There's another important place where we use ATP, and that is to actively pump calcium out of the cell. Calcium is an incredibly important intracellular signal in that we have so much outside of the cell that we can rapidly increase the intracellular concentration by allowing it in, and it's this very powerful signal. So in this case, if we don't have enough ATP to continue to pump calcium out, calcium is going to flood down its concentration gradient, and we're going to see this big increase of intracellular calcium. Now, this is a big problem because when calcium goes up, it is a signal for enzymes within the cell. Specifically, we see the activation of phospholipase. Phospholipase is an enzyme that's going to attack the membrane, the phospholipid bilayer. So we're going to see it kind of start chipping away at the membrane. It can start attacking the membrane of the mitochondria. It could even attack the membrane of some critical organelles, such as a lysosome, which is also phospholipid membrane bound. Another enzyme that gets activated is protease. Protease, as the name implies, is an enzyme that breaks down proteins. So this cell has an intricate cytoskeleton, and protease is going to come and start breaking that down. When you break down the cytoskeleton, the cell begins to lose its shape. So this swollen cell now kind of starts to form blebs, and there's no longer this nice structure to the outer wall because nothing is anchored to the cytoskeleton. The next enzyme that gets turned on in relation to the high calcium is endonuclease. Endonuclease is going to be an enzyme that targets the DNA specifically, the nucleic acid, and it's going to start chipping away at the DNA, the blueprint of our cells. The last one on the list is ATPase, which is an enzyme that breaks down ATP. So we see kind of a vicious cycle here where we turn an enzyme on that's going to break down even more ATP. So at this point, with the way the drawing is laid out here, this cell is still in reversible injury. Certainly things aren't looking good, and this sick, dysfunctional mitochondria is starting to produce reactive oxygen species, which are highly reactive molecules that can damage pretty much anything they bump into, right? They can damage membranes, they can damage the proteins, they can damage the phospholipid membrane. And as they accumulate, as we get more and more, we start to see the state of oxidative stress. Oxidative stress is when you have an accumulation of free radicals that are now causing accumulated damage that can significantly affect the cell. So the presence of our enzymes that were turned on during reversible cell injury is significant because they're still on. That calcium is still elevated within the cell. In order for that kind of reversible injury to have gone into reverse, we would have had to restore oxygen so that we can pump that calcium out, which would have turned the enzymes off and allowed for healing to occur. But in this case, the oxygen has not been restored, and so the cell is continuing with these enzymes that are active. So phospholipase is going to start to create, whole gaps in this membrane. We're going to see, sections just kind of float away because we've broken through the membrane. Once you no longer have an intact cellular membrane, this cell cannot survive. We see a loss of the membrane of the nucleus. And as we have longer and longer times for endonuclease to be present and reactive oxygen to be present, we end up ultimately losing all of our nuclear structure. Without a blueprint, this cell can never survive. A big part of these late stages of cellular injury is a very sick, unhappy mitochondria. It was already producing reactive oxygen species. That process would only get exaggerated as the cell becomes more and more dysfunctional and that mitochondria becomes kind of leaky from the membrane damage. We know that the membrane has kind of kill signals within it, where if it gets leaky enough, proteins from inside of the mitochondria can spill into the cytosol. So an ill, kind of sick mitochondria is a key part of this. So we talked about the loss of the integrity of the membrane of the entire cell, but we see the same thing in some of our organelles, and specifically we'll mention lysosomes. So lysosomes, as they get attacked by phospholipase, digestive enzymes can spill into the cell, and they will eat up and destroy anything in their path. So that creates a huge problem for the cell that's not able to come back from. In addition, we see further damage to proteins, complete loss of DNA, damage to mitochondrial DNA, and the cell at this point cannot survive. So the intracellular elements will spill out into the interstitium around the cell. The presence of intracellular organelles and other things within the cell in the interstitium will trigger the inflammatory process. So cellular necrosis leads to acute inflammation. So this entire process is a continuum in that the natural effects that happen when there's not enough oxygen to deliver to a cell. When we're still at the stage where there are enzymes that we could turn back off by getting rid of the high intracellular calcium, when we can replenish or return our sodium gradients to where they were, we can move that fluid back out. But once we have damaged the cellular membrane, once we have damaged the nucleus and we've damaged the blueprint of the whole cell, the cell can never recover. When the mitochondria becomes too dysfunctional and leaky, the cell can never recover. Once we have digestive enzymes exposed to cellular elements, the cell cannot survive. So when we can still turn things off and replenish what we have, it's reversible. As soon as there are steps that we cannot turn off, we cannot undo, the cell is an irreversible cellular injury and undergoes cellular necrosis.